There we go. Yeah, so the first question we wanted to talk about is we're starting to get a lot of exciting data from some of our different sponsored projects. A lot of this is high dimensional data, so RNA-seq data, tons of information there. And so there's a lot of potential ways that we could analyze this data, compare between data sets, between individuals, between samples. And so there's a lot of potential, and we'd like to get sort of your feedback on, where we go from here with these different data sets. So some of the data sets that we have in hand currently are RNA-seq from 8p and trisomy 21 fibroblasts. We have the data that Joe shared with us from RareBase, the RNA-seq of the glutamatergic neurons. And we have the data that Alison Moultrie shared with us a few weeks ago on brain organoids, the single cell sequencing data. So I guess we'll open it up to the floor to folks.
And yeah, I think we can get into the details of those data sets. That's something we definitely feel free to kind of bring up as fair game. I think the RNA, the fibroblasts and the iNeuron data sets, I believe, are bulk RNA-seq, whereas the organoid data set is single cell. So I think we can kind of get into the particulars of all that. That's definitely fair game for discussion. So, Nicoletta, feel free to raise your hand, maybe, unless you want to just unmute and chime in. We can have a raising of a hand process as well. But Nicoletta, please.
Hello, thanks everyone for joining in today. Our project is focused on the fibroblasts, and I think that looking at cellular phenotypes that may be targeted pharmacologically, it would be good to have a bit of knowledge about the pathways that may be differentially regulated in 8p lines versus controls. So perhaps if we can access this data, that would be useful. So what is the extent of the analysis that is available? Do we have, what is it available? What are the cell lines? What is the extent of the analysis that has been done? Is there anything else that needs to be done? How can we progress from there?
Yeah.
So I'll chime in just first. We're running our own kind of reanalysis of the fibroblast data set. There was a kind of very sweeping sort of analysis done initially. This data set was from Angelica Ammon's lab, so several years old. And that's, I guess, maybe some other caveats that might be needed to be mentioned. But so we're hopefully this weekish, we should have kind of our own independent reanalysis for gene enrichment, geo enrichment stuff, kind of the obvious things to sort of look at that we could sort of try to line up with your observations that are kind of more image cell phenotypic data. But yeah, Whitney, maybe you can either describe just sort of like how people can check what is the exact line, what identity of that line, where, for example, how does that relate to the OCORD data set? Like maybe you could speak to those issues as well, so that when you can share out this information, and you can maybe also speak to like where the data sets live in the cloud.
Yeah, so currently we have the data, raw data transferred to an S3 bucket that we can make available to anyone who wants to look at that data. The specific lines that we have, there is a manifest along with the data, and the lines are named in the same way that the fibroblast lines that we gave to you are named. But we can also, I don't think it's currently linked, those IDs aren't linked with the OCR data or patient IDs, so we could certainly do that.
Okay, thank you.
I think two of the lines that you have, Nicoletta, we have RNA-seq data for, and then I think you have maybe two more that we don't.
Okay, thank you.
Yeah, anyone else have any thoughts on either one of those, something about the specific model in which the data set was generated, or just a comment on how one might do these kind of a meta-analysis of RNA-seq data, given that these are, very different types of models, two-dimensional cell models versus three-dimensional, more complicated, organoid arrangements. So yeah, anyone can feel free to sort of comment on any aspect of how we might be doing this analysis together.
We have phenotypic data for some of these individuals. Sharing that data, generally is a little bit more tricky, but if you want access to that data, we can send you a form to fill out so that you can see phenotypic data for not just those lines, but all of the lines that we have in our biobank. So feel free to reach out to me for that.
So I think it's a pretty big question. Oh, sorry, we have a question in the chat, I think.
I think.
Oh, okay, that's from earlier.
Yeah, it's from, go ahead, please.
So anyone who's done RNA-seq analysis can tell you that there's 12 ways to sundown to actually do this, right? And a lot will depend on, I think what would be really helpful is, for example, you just mentioned the fibroblasts, and that they're from, this is an overlapping panel with the, taken from the Okur et al. Cohort, is that correct? So
Yeah.
that could be really useful. I didn't realize we had that. So the most important thing you need is a table. You need a table for the different samples, the different batches that were run, how many replicates you have of everything. Like compiling that data will actually give us an idea what we can do with it.
Okay. Great suggestion. Thank you. Any other thoughts on this topic? Otherwise, I think maybe we can open up discussion on the second front that we kind of prompted.
I was curious whether Harui or anyone has thoughts, because in parallel with the 8p fibroblast sequencing, there's also trisomy 21 sequencing that was done. So curious if folks think there's opportunities to do some comparison there, whether that's worthwhile or not.
Yeah, I think that's one of the things we're working towards. We still haven't generated any of the iPSC or MPC data, but once we do that, the bigger picture of my lab is trying to do beyond just chromosome 21 and other CMVs. So we're generating trisomy 18 iPSCs and MPCs. We've gathered basically every trisomy or aneuploidy lines with the exception of chromosome 1. So we're slowly but surely working through those. We have Pitt-Hopkins syndrome, and we're trying to get our hands on some DUP15Q lines. But the bigger picture is to try to compare all of these at the same cell types, right? iPSCs and MPCs. So we've never done any fibroblast work. So we have no fibroblast data. But for trisomy 21, there's a lot of fibroblast RNA sequencing data. So if necessary, I can assist you in getting the data sets and putting it into your pipeline and comparing how fibroblasts behave. There's one really great study. Actually, there's a bunch of great studies, but there's this monozygotic twins where one has Down syndrome and the other one does not. And that paper has really great fibroblast RNA sequencing data. So maybe we could start there. And then I don't know, Stefan, maybe you have some RNA sequencing data from DS, right?
Yeah, so I think I'm speaking next month. And I figured I would talk a little bit about our trisomy 21 work as well, and give an update on that. And I want to kind of point out what we learned from that work and from our Turner syndrome work, and how we plan on applying it to the 8p work. So I just wonder, so you're interested in commonalities between Down syndrome and 8p because of the neurodevelopmental phenotypes, although some are not really overlapping, like in terms of epilepsy, etc. So
actually, there is a higher incidence of epilepsy in DS as well, right? It's just that the numbers are so big in terms of the number of individuals with DS, and the incidence is lower.
Yeah.
But there is, and then if you know, the mouse model, some of them have spontaneous epilepsy-like features.
Yeah.
So there is some epilepsy in DS as well.
Yeah.
All right, if not speaking, if you can please mute just to reduce the background noise. Thank you.
I was wondering about. It's not me, I promise.
No, it's not you. No, we're good. It's gone.
I was wondering about these fibroblast lines. Do those come with parental lines as well?
No, we don't have parental lines for any of those, unfortunately.
Okay. I mean, you know, just because Hirui mentioned so we work a lot in isogenic models specifically so that we can exclude the impact of other variation. And so monozygotic twins obviously are fantastic for that. And of course, the next best thing, as you guys know, are the parental lines. So that's, and I know we have those for some of the iPSCs. Could you actually give a brief update on the iPSC generation, like in terms of, I know, so the pathfinding trio is available. Are there any other in the works currently?
Yeah, and they're almost completed. They're currently finishing up the karyotyping, so those should be done in the next couple of weeks, so we can share those. To your point about isogenic controls, though, one of the fibroblast lines is actually a mosaic line. So I don't think it's, you know, the separate genotypes have been isolated, but that's a potential. And we are creating iPSCs from that particular fibroblast line, so we'll have both the variant line and the control.
Brilliant. And are those, is that an inf-dubdel case?
I believe so, yeah.
And is it male or female?
It's a male.
Okay. Very cool.
Yeah.
So that's typically what we would start with. That's how we went about the Down syndrome work, is from a male mosaic that we generated. Well, I'm going to talk about it next month, so I don't have to now.
Cool. Any other thoughts here? Because I think this is exactly the kind of discussion we wanted to catalyze here. If not, I think it maybe segs into our next topic area, which was sort of accelerated aging, and so maybe thinking of, again, the experience with trisomy 21 and whether that sets kind of the priors for AP or not. I don't know, Whitney, do you want to kind of formulate the way you had put it in the email?
Yeah, so our question was, how do we assess accelerated aging in AP heroes and in AP disease models? So we don't have a lot of data on, you know, how this syndrome progresses or what happens in older AP heroes, but we want to start thinking about them now. And so, you know, what can we be doing now to try and, you know, assess, you know, what we should be thinking about for their future. And I think Veena can probably weigh in on, you know, some of the information she has about some of the older AP heroes that she's familiar with.
Yeah, and I guess it's not many. And I think Katie's also on this call, and she's our patient engagement manager now and actively talking to families every week. And trying to reach as many as possible. So, you know, it's a little, we have some starting, you know, clinical phenotypic data. It's not, I wouldn't say it's like longitudinal. We are asking for like background medical records and things that we need to curate, honestly, and go through. But, you know, it's, we're just kind of trying to right now think about, like, you know, how do we ask the right, just big picture to, so I'm trying not to prompt another question, Ethan and Whitney, but it's in my mind. It's like the bigger question of what's the framework, so, like, what is the scientific framework and the methods we need to kind of determine high risk factor for, you know, adult onset. And you can imagine, you know, fill in the blanks. Right? We're kind of seeing anecdotally a little bit of everything, you know, that you read about just generally in your papers. So, you know, whether it's some severe mental health conditions, which, you know, is like that big neuropsychiatric bucket that is part of the AP roadmap. And, you know, we definitely want to pursue better understanding that in the AP syndromes. So right now we're just like, you know, anecdotally starting to come up with questions and then determining how can we really do this in a systematic way. And then ideally intervene right before it happens. If we start learning that things are happening in the late 20s, things are happening in early 40s, you know, we have very few cases of that. So it's hard to determine in like the humans, but how do we do that in our in vitro models, for example? I know I prompted a whole different question.
No, that's great. This is, I think it's all embedded. It's just, yeah, putting, I think, a more, you know, real world perspective on it.
Well, Cheryl's on the line too. I don't know, Cheryl, if you wanted to add.
Yes, I guess this is my area that I'm very interested in. I'm a parent of Hannah, an AP hero, and she is actually 30 years old, maybe one of the oldest ones, but there are some that are older. And she has experienced a lot of aging related medical conditions lately. And, you know, like she has severe osteoarthritis in her feet and ankles. She has low sodium now, low blood pressure. She also has recently been diagnosed with aperistalsis of her esophagus. So, you know, I asked if they've seen anybody else have this, and they said it usually happens in older people. They've never seen it in someone that young. So, you know, for me, with my daughter's age, anything we can learn from this, and I'm willing to do a skin biopsy on her if that would be of benefit, because you know, anything that would benefit her or any of the younger people, I'm certainly willing to do.
I mean, just to echo or amplify that, we saw talks at the meeting about a direct differentiation of neurons from fibroblasts, where you wouldn't have a resetting of the epigenetic clock, and wondering whether we should, you know, is this exactly the, maybe this kind of pathfinding, you know, skin sample that could start to shine a path. But yeah, anyone can, you know, Rui, if you want to jump in.
I think maybe even taking, you know, trying to do an epigenetic clock sort of experiments initially before generating any of these directed neurons or what have you, is just to see, is there a change in, you know, the methylation profile of the DNA? So I think maybe we can add it, you know, add Hannah samples to the other AP samples that we have, and maybe this could be a, you know, a longitudinal study, but in multiple different individuals from multiple different age groups. I think that would be beneficial to start with, and you know, also the speed at, you know, how that experiment would go would be much faster than trying to convert it to, you know, other cell lines. I would think, I don't know. And Stefan has more experience than I do, I feel.
Well, I just added the paper that Hirui is referencing, the epigenetic clock. It's a classic paper for essentially using DNA methylation data to assess age or cellular age in a quantifiable, reproducible manner. And so this is Stephen Horvath's work. He ended up actually being scooped up by L2s, which is the anti-aging startup. I'm not even sure I'm pronouncing it right, but so we haven't actually, so we've done a lot of DNA methylation analysis. We haven't actually looked at his DNA methylation clock ourselves yet. We'll do that, for example, with our Turner syndrome lines at some point. I think that's a good starting point. One of the things is sorry, I'm just recapping things that you guys have already said. But Ethan mentioned that the clock would be reset when you reprogram to iPSCs, and so that's correct. So that's one of the things that's shown in this paper. And so, but on the flip side, if you differentiate, I'm just thinking out loud, if you differentiate direct to neurons via NGN2 from fibroblasts, then you have the biopsy date is basically then is basically like a confounder, right? Because you may have, you know, the neuron may not behave well either because there's accelerated aging that you've captured, or it may not behave well because you've taken it from an older patient and you're comparing it to a younger patient. So then you'd need age-matched controls that, you know, healthy age-matched controls, which you may have in your fibroblast bank. But the other way of doing that would be to reprogram iPSCs, have them the same starting age, and then see whether they do age in an accelerated fashion, right? When you differentiate them. But since you already have the fibroblast lines, I think the most straightforward thing is, and I think that's what Hirui was getting at, the most straightforward thing is to get DNA methylation profiles for them and then compare how those DNA methylation ages compare to the chronological ages at biopsy. Because then you already have preliminary data that might show you that there is accelerated aging.
Can you do that on blood?
You can do that on blood. You can do that on skin. I don't know what I'd have to look back at this paper because it's been a while. I don't know
What
what
about hair? It's come up.
It's probably not a lot of cells, right? But you could culture probably from, but you know, since you already have a fibroblast bank set up, I think that would be, I would probably start there, because with blood you can have funny clonal phenotypes, right? Because with aging, go ahead.
No, I just wanted to add, so for our mosaic Down syndrome individual cohort, Colleen Jackson Cook has been doing, you know, methylation aging for those individuals from blood samples. So this has been going on for, you know, 3 decades at least that she's been doing this. So maybe she's a good person to talk to who has extensive knowledge of, you know, working with fibroblasts, blood samples. She does cheek swabs. So maybe.
You know, it's funny because our partnership with Illumina, they did say they were going to run DNA methylation on all the, you know, extracted DNA from the blood samples we provided them. So I'll also ask them. That would be at least like 12 AP heroes, mostly in version Ductel, but and younger. So I'm not sure what that'll show us. But would you say that then, like, just thinking systematically and designing this is like we would try to maybe for this DNA methylation study to try to recruit samples from older AP heroes, right? Like above age 15?
I mean, I think ideally you just want to collect anything you can find, right? And then based on that, on the ages that you have, you can sort of do from very young to the very oldest that you have in between. And then I think maybe that can give us a trajectory of how things are going. But, you know, there's going to be individual variations, right? And what Stefan was saying with the iPSCs is that in vitro, you can reset everything to the beginning and then see how things go in time, right? If there is, you know, that CNV-induced acceleration of aging, you may be able to capture it by resetting it back to the beginning.
Okay, and this is Colleen Jackson.
She's at Virginia Commonwealth University.
Okay.
I'll send you her information. I can introduce you.
Thank you.
Yeah.
The other thing.
Here we side note, we have 215 samples.
Oh, wow, that's a lot, actually.
Yeah, so Stefan's part of that commission work early on too. So we had blood samples. So in case you, and some iPSCs. So if you wanted, just I'll introduce you and connect you. And I feel like, don't you have them at UConn too? Like, I don't know, in the core?
Yes, we do.
Yeah, okay.
So those were generated by Stormy Chamberlain's lab, who got a job at as head of neurogenomics at Roche. But the lines are still here, and I think you can still request them. So we have a person sending them out, and they're very well characterized lines. I haven't worked with them myself, but they're here. Hiroi, send me an email.
Okay, we'll do.
Nicoletta?
With fibroblasts from older heroes, we can also look at the other markers of aging, mitochondrial function, and DNA damage accumulation with a matched control for aging. So age matched control and 8p lines. These cellular phenotypes are more likely to show if there is an aging phenotype in these older heroes.
But we haven't looked at that yet, right?
No, the lines we have are from young
Yeah.
heroes.
Okay.
All right, this is also a nice conversation segment. Anyone else want to continue or amplify a point? I think definitely.
Perhaps the other assay that would be good to do in correlation with the methylation would be a release of cytokines that are markers of aging, SASP, GD, DDF15, some of these. But yeah, in addition to methylation.
Do you guys want to start a whole study on this now? Because I'm asking because the phenotypes that that were described, so they didn't sound like typical, for example, progeroid phenotypes, right? That you would have in classic accelerated aging conditions. I think the comparison with Down syndrome is probably a really good one, because it's pretty well described in vitro and in animal models, as well as in probands. So, maybe we should, if you guys want to look at that, we should probably look at phenotypes that have been demonstrated for Down syndrome. And Hiroi had this very nice work in Cell Stem Cell about senescence. Hiroi, did you see a correlation with between your senescence phenotype, so the secretory senescence phenotype, and the clock?
We actually never really compared it to the aging clock per se, but that's something we can do. There is a SAS profile, but it's different than the SAS profiles traditionally observed in the aging brain. There are some cytokines, but it's mostly MMPs. So, matrix metalloproteinases. So they're involved in extracellular matrix reorganization. It becomes tricky because there's also, developmental senescence, which I think what we observed is more similar to that than aging associated senescence. So we don't really know. But that's what we're hoping to start to see with this RNA sequencing and MPCs.
Thank you.
Yeah.
Do any of the family members here want to just ask questions? I mean, anything honestly about your AP Hero want to share with the researchers? Just anything that comes to mind, I think this is why we're here together.
I think it's just also pointing out, the relevance of, getting kind of foundational data sets in motion now, or collecting samples now, anticipating a longitudinal study, and just realizing that takes a true partnership between the researchers and the families, and kind of being very proactive about something that might require many years to sort of flesh out in the data. But I obviously it kind of starts with baby steps. But anything else people can be thinking about to get us thinking, going off on the right track, please raise those points now. I think this has otherwise been a great discussion in terms of getting researchers and families on the same call in the same lab meeting and kind of sharing observations and sharing insights really. So I think this sort of open forum, I think, served its purpose. So thanks, I think, for everybody kind of rolling with the schedule change here. I don't want to cut the conversation short, but if we've sort of exhausted the topics of kind of RNA-seq analysis and accelerated aging, which I think we had great conversations on, we can kind of wrap here, or we can have some concluding remarks from anybody. Yeah, please.
I was just gonna say one more thing. So for Down syndrome, I think what's held Down syndrome back for a while was in terms of like getting enrolled in clinical trials, right? Was that there wasn't a natural history study, and so Lumine, for example, has been working on putting that together. NIH has started doing that as well, and so that's going to be even more challenging with a more rare patient community, right? So I wonder whether essentially extending kind of what you started with the Okur et al. Paper, extending that and turning that, like following that cohort and turning that into a natural history study. Maybe I think it's probably in your plan already, right?
Yeah. We've been doing it. We've been able to collect with some families because part of what Katie and I have been trying to talk to, and it's people enroll, they may have participated in that, and then you kind of think like that's it. And so what we've been trying to share with Wendy Chung as still our scientific advisor is let's go to year 2, year 3, year 4, like we're going to have to do this annually. And so most of those families, at least I think 60-70% of the families in that paper have now reconsented in the AP registry as a longitudinal study. And Dr. Chung has also transferred the data over to us too, so that we can kind of bridge and connect that information over time.
That's great.
Yeah, but we haven't actually gotten to a point where anyone is looking at patterns and doing any visual analytics on it yet. It's not enough data points yet, but we're also building the skeletons right now of a portal that not only just dumps data, is encrypted and secure, but actually layers in some, I would say, like, basic visualization tools that can start popping out for all of you to access. So you can do little cohort analyses and we'll continue building that together as a team. Like, I wish I could see this. I'm working on this, my NPCs are showing XYZ, and I wonder if there's, in the MRIs, it's showing this information, or the EEGs picking this up. Do you have that? If we don't, then we also can go back to the families and start making those connections as well. So this is very much a partnership, but we're trying to think ahead of what you'll all need soon as you start generating some prelim data in the lab.
That's terrific. Thank you.
But we need this kind of advice and guidance from all of you. So please, like, everyone's, questions or some paper that you've seen, and, an investigator that you might know that could help answer some questions on AP Heroes, make those introductions. We're growing our medical and scientific advisory board because it's been a little informal to date. And, and so we understand you're working on a lot of other disorders, and it's not just AP, but very much open to that. We need to hire a few people on the foundation side to understand how to really build, like having an epidemiologist maybe like on the team at some point to really look at the natural history data, and people that have worked at various institutes at the NIH, maybe in the Down syndrome, that are looking to do some part-time consulting with us. So again, like, very open, and just please, feel free to give us your guidance. I mean, that's what we have these roundtables for too. So this was kind of nice today because it wasn't, just presentation, a short time for conversation, but very helpful. Yeah, I'm coming.
I think on that positive note, maybe we can wrap, and this is available for review here. I definitely think we will review. Whitney will kind of hopefully summarize some of the points that were brought up. I felt like we achieved a lab meeting feel here, and maybe the researchers will agree with me, and the families, if you don't know what we're talking about, that I think you'll kind of appreciate that. This kind of meeting of the minds here, where everyone just sort of is contributing and everyone can contribute, is sort of the spirit that we've tried to achieve here. So thanks everyone for pulling together here and making today work out. So have a good rest of your day, and also please make sure, if you're not subscribed, I'll do a little plug. If you're not subscribed to the Chromo 8P Substack, you should check that out. It's where a monthly newsletter goes out that kind of is the companion to this roundtable. And there's a highlight of the roundtable included. So if you're not subscribed, please do. We'll include a subscription link in the email as well. If you are subscribed, thank you for subscribing. And if you have any content ideas for us, please let us know. We're going to try to hopefully put out more posts that talk about RNA-seq data as we start to analyze it, and let the data sort of live free, especially on the kind of preclinical side. I think we all agree that can only do better if we put the data out there, and not just to have everyone analyze it, but as a way to kind of entice others to come into our community as well and help us grow. So thanks everybody again for joining the April Research Roundtable, and we will see you again next month.
Thanks, everyone.
Thank you.